Hello from Amboseli. My name is Jenny Tung, and I am happy to be talking to you today about some of my group's work on the social environmental predictors of accelerated aging in wild baboons. Thanks very much to Hannah, Julia, and Alex for organizing this session and inviting me to participate. In this case, I'm glad it was virtual because it's giving me the opportunity to talk to you while I'm in the field with the baboons themselves. So the work I'll tell you about today is predicated on the fact that while chronological aging is the same for all organisms, the rate of physiological decline and therefore the increase in risk of mortality is not. In other words, although an animal that lives to 10 years of age has experienced the exact same number of seconds, minutes, hours, years as another animal born at the same time, that animal may have the mortality risk of an individual years older, or alternatively, an individual years younger. These differences in mortality rate and physiological decline are often explained by systematic factors, including genetic makeup and aspects of the environment. This is extremely well studied, of course, in our own species, in which differences in the environment, exposure to cigarette smoke, for instance, alcohol consumption, past history of disease, such as cardiovascular disease, can have major effects on the probability of mortality at any given age. What I'm showing you here is a summary of exactly those types of environmental effects on the mortality risk of men and women, adult men and women, in the United States and several other developed nations. And the further the bar is to the right in this figure, the bigger of an impact the environmental effect on the left has on mortality risk. Not coincidentally, this particular analysis highlighted the importance of aspects of the social environment, particularly social support and social integration, in defining differences in mortality risk and hence differences in biological age for individuals, even if they were at the same chronological age. The effects of the social environment on health, mortality, and fertility are of great interest to the work that I and my colleagues do at the Amboseli Baboon Research Project in Kenya, which is where I happen to be right now. This is a picture of one of our field assistants, Kinua Waratere, in the field watching the baboons. And this is a typical way we've collected data in Amboseli now for 50 years of continuous observation. I'm fortunate enough to do this work in collaboration with my colleague Beth Archie at the University of Notre Dame, Susan Alberts, who's also at Duke, and Jean Altman, who was one of the founders of this project in 1971. In Amboseli, aspects of the social environment and the particular ecological circumstances of individual baboons have quite pronounced effects on the likelihood that they'll survive to old age. And this is of great importance from an ecological and evolutionary standpoint, because survival to old age is one of the most salient predictors we know of lifetime reproductive success. In other words, the currency of Darwinian fitness. This is shown here in a figure from a paper we published a few years ago, in which we're looking at age at death for females in this population after achieving sexual maturity. And you can see there's a quite strong linear pattern between survival and the number of offspring a female leaves behind. Understanding why some of those females live quite long and why some of them make it barely out of sexual maturity is, again, something that we are trying to understand in much more detail. But to do that, we need more information than we can gather from mortality itself, for a few reasons. One, mortality only happens once, so it doesn't tell us anything about the dynamism of biological aging over time. Two, we only have mortality data for a subset of individuals. We don't know when individuals currently in our population will die. And for some individuals who have lived in our population in the past, we lack death records. And third, because mortality is a combination of those systematic environmental processes and stochastic age-independent mortality risk. So what we need to do is go from simple measures of chronological time, represented by that clock I showed you earlier, to measures of senescence at a biological level. And to do this, we recently turned to a concept called the epigenetic clock, represented here by this clock made up of actual DNA strands with little marks on them. That representation comes from the fact that one of the crucial epigenetic marks that contributes to gene regulation in baboons and other vertebrates is a mark known as DNA methylation, which is simply the addition of an extra methyl group to cytosine bases in mammals like us and like baboons that tends to happen most often, where a cytosine base is followed by a guanine base. These chemical modifications can be present or absent at particular CG motifs in the genome, and they can differ in the proportion of cells, of the proportion of DNA strands on which they're present in different tissues. It turns out that work by Steve Horvath and others from 2013 shows that as tissues age, they also change in their level of the addition or subtraction of these methyl marks at particular diagnostic sites in the genome that together produce a highly accurate composite measure of chronological age. And you can see the results from that early work in humans shown up here on the right-hand side. There's a highly linear relationship between predicted age based on DNA methylation levels at these diagnostic sites and actual chronological age in individuals. Since then, there's been a variety of work showing that DNA methylation age or epigenetic age also predicts mortality risk, time to death, and that is in turn predicted by environmental variation that we know has consequences for mortality. So we decided to construct a similar clock in the baboons that we study in Ambicelli. We can do that because while the majority of our work is observational, occasionally this is actually why I'm in Immobilization right now, we temporarily immobilize known individuals from our population, individuals, for example, with known birth dates and therefore known chronological ages, in order to collect blood samples and other types of biological or morphometric data. It works basically like this. This is again our very talented field assistant, Kinyo Waritere, in the field holding about a meter long metal tube in his right arm. You can actually see it vertically in his right hand. He's waiting for an opportunity in which a known individual in our population is not paying attention to him and no other animals in the population are also looking. When he gets an opportunity like that, he'll lift up that metal tube, the blowgun, and breath propel an anesthetic containing dart towards the target animal, who then falls asleep, allowing us to collect those samples that I mentioned earlier. Afterwards, those samples are taken back to the United States, DNA is extracted, and we can perform a series of analyses, including calibration of an epigenetic clock. This is work that was led by my graduate student, Jordan Anderson, and my postdoc, Rachel Johnston. Okay, so what we did was extract DNA from 245 unique baboons who have lived in our population and for whom chronological age is known to within a few days' error. We then performed a technique called reduced representation bisulfite sequencing, which allowed us to profile methylation levels on a genome-wide scale in each of those DNA extracts. Ultimately, after quality control, it allowed us to measure DNA methylation levels, which range from 0 to 1, 0% methylated to 100% methylated, at about 450,000 CpG sites genome-wide. Just for context, there are about 20 million or so CpG sites in a mammalian genome, like the baboon. We then used a machine learning method called the elastic net to ask whether we could identify a subset of those sites that were strongly predictive of chronological age. And in doing so, we identified 593 age predictive sites that could fairly accurately give us a measure of chronological age, give us a predicted measure of chronological age in samples that weren't involved in the original training set. So this is what our predictions versus known chronological ages look like in female baboons on the left and in male baboons on the right. As you can see, in both cases, we get a highly significant relationship between known chronological age on the x-axis and predicted chronological age in years. In females, these measurements are a little bit biased, so we tend to underestimate the age of older individuals and overestimate the age of younger individuals. In males, the clock ticks much more regularly according to chronological age. The x equals y line is that dashed line, and the best fit line are the solid lines in each case. In both cases too, if we take the subset of our data where we have longitudinal samples, where we know the change in chronological age between those sampling dates, predicted age also follows that time interval. So in other words, chronological age between two dots connected by the same line that represents the same individual tends to go up with time, and tends to go up with time in a way that is tightly correlated between predicted and chronological age. Okay. Just to give you an idea of the relative performance of the epigenetic clock in predicting chronological age, here I'm showing you a comparison between epigenetic clock, overall variance explained, bias, that is the degree to which it's off from that x equals y line, that perfect prediction, and the median absolute deviation per individual in years. The epigenetic clock is shown on the left-hand side for males and females respectively, and other measures that can be used to predict age, dentine exposure, that is tooth wear, body mass index, and cell type proportions, either from flow cytometry and blood smears, are shown as a comparison. In general, the epigenetic clock, especially in males, performs as well or better than any of these other measures in getting us accurate predictions of chronological age. That being said, of course, in this population, we have a longitudinal field population in which we actually know the ages of these animals when they were born. We don't really have a need to directly predict chronological age, but we're quite interested in when chronological age isn't the whole story. That is, when some individuals appear to be biologically older than they actually are, or biologically younger. So here in particular, we're trying to ask about environmental predictors of when individuals are predicted from the epigenetic clock to be significantly older or substantially older than their actual age, or those cases where the deviation is in the opposite direction, where they actually look much younger than their actual age. We focused on several of the most important socioecological predictors of mortality rate known in this population, including the rates of early life adversity. So in this population, individuals who experience no particular major forms of early adversity, being born to low social status mothers, socially isolated mothers, being born in a drought, and so on, live a median of 10 years longer than individuals who experience 3 or more of those sources of early adversity. So this is a major predictor of lifespan in our population. We asked about the effects of social bond strength, a measure of social integration or social support. In this case, we looked at this specifically in females because we simply didn't have the sample size where we had these measurements available in males. But in both males and females, individuals who have weaker social bonds tend to die earlier. And this is on the scale, again, of years. And we also looked at the effects of dominance rank or social status. In Ambicelli, high-ranking males actually tend to have a slightly elevated risk of mortality, whereas females don't seem to be affected, at least for that outcome measure, by their rank. I'll cut to the major results, which is that of all of these predictors, only male rank predicts accelerated epigenetic aging. So here I'm showing you ordinal dominance rank on the x-axis for males. High-ranking males are on the left, low-ranking males are on the right. And relative epigenetic age, that is whether they are predicted to be old for age or young for age by the epigenetic clock on the y-axis. We see a strong negative correlation, meaning that high-ranking individuals tend to be predicted to be older than they actually are, and low-ranking individuals tend to be predicted to be younger than they actually are across these 105 samples in our data set. This also translates to longitudinal patterns of aging. For a small subset of males, we had samples taken at 2 different times in their lives, separated by several years, in which they also changed rank. Animals who were sampled at a higher rank tended to look older for age than when they themselves were sampled again at a lower rank, and vice versa. So the arrow here is in the direction of the temporal change over time. And you can see, by and large, these all exhibit the same type of negative relationship we observed in the cross-sectional data. In fact, this may explain some cases where in our longitudinal data, despite time passing, 2 of our males actually looked like they were younger by epigenetic age predictions than as chronological time passed. Those were actually animals who actually fell in rank between their first and second samples. So these sites, these 593 sites, What do they do? Are they functionally relevant, or are they passive biomarkers of aging? Well, we know by intersecting data from this analysis with data from other types of analyses that CPG sites in the clock are enriched in empirically identified enhancer-like elements. That is, they are likely to be involved in gene regulation and be functionally relevant to the physiology of these animals. In addition, we know that CPG sites in the clock are more likely to occur in regions of the genome where individual sets of sites change their DNA methylation status with age, perhaps not very surprising. However, one of the particularly interesting things from our analysis is that we found that CPG sites in the clock that tend to positively predict older age also tend to occur near genes that are upregulated upon experimental immune activation. Conversely, CPG sites in the clock that predict younger age, also tend to occur near genes that are downregulated upon immune activation. Together, this evidence strongly suggests to us that these CPG sites are not simply passive biomarkers, but are probably tracking something physiologically important in the animals that tends to change with chronological age faster for some individuals than others. Recently, we were able to pair these types of data with additional data that we gather from darting. So this is a picture of a blood draw during darting, where one of the experiments that we do is to draw blood samples into paired tubes that contain cell culture media, basically the minimal ingredients required for those cells to live outside the body for a short period of time. We draw blood into tubes that contain media only, and blood into tubes that contain media spiked with an immune activating reagent, in this case, lipopolysaccharide, which mimics infection with a gram-negative bacteria. We incubate these tubes in parallel. Here's our field incubator, which keeps them at 37 degrees C, body temperature, and then we harvest those cells for gene expression analysis downstream. We've now done this for a number of individuals, including a large number of males in our sample. And what we find is that dominance rank is a significant predictor of gene expression levels for many of the genes involved in regulation of the immune system. And in particular, what we observe is systematically higher expression of genes involved in the inflammatory response and in the innate immune response, including these key antiviral pathways, for males that are high ranking in our population versus males that are low ranking in our population. Each of these distributions represents somewhere from about 50 to a few hundred genes involved in the pathway on the left. This is particularly interesting to us because it is actually diametrically opposite from the pattern that we pick up from females who are being sampled at the exact same time, where in that case, it's low-ranking females who tend to upregulate these immunomodulatory genes. So we think this might be explicated by the fact that baboon males have to physically compete for rank. In order to get to a high-ranking position, males have to be able to win fights with other males, while females actually insert into the dominance rank hierarchy based on the ranks of their mothers. It's a nepotistic system in which rank is very stable over time. Males who lose body condition fall in rank. So they occupy a dynamic rank structure over time where they tend to increase until they hit peak condition about age 10 to 12, and then fall thereafter as they age. So that elevated level of inflammation may in fact contribute to the physiological aging process, but be valuable for those males because those exact genes are the types of things that are important in wound healing processes, which are risks they can incur in this type of behavior. So this is from a paper by my colleague Beth from 2012 that shows that high-ranking males in the solid line tend to heal faster from comparable injuries than low-ranking males. So this may be a type of physiological change that we're picking up as a biomarker of senescence that is also functionally important for males who are occupying that particular position in the social hierarchy at the time. I want to point out that may mean cost to health later on. We think this is in fact a cost to competition for males, but it's probably worth it from the perspective of the males themselves. The best predictor of producing offspring in male baboons is achieving high rank. So here we may be observing a trade-off between maximizing fitness and minimizing physiological decline. So in sum, despite the fact that we have multiple social predictors of lifespan, not all of those predictors of lifespan are good predictors of biological decline, at least as captured by epigenetic aging in peripheral cells in the immune system. Additionally, the fact that we see longitudinal changes in this sort of accelerated versus decelerated epigenetic aging process as males change rank lends support to the idea that what we're capturing best through these measures are recent effects of the environment, a recency model for social environmental effects on health, in contrast to longitudinal biologically embedded effects of, for example, early life adversity. Third, these data, in combination with other information on the baboons, suggests that markers of biological senescence may in fact be highly sex specific, at least when it comes to environmental predictors of accelerated or decelerated decline. In this particular case, the epigenetic clock or variation in the epigenetic clock relative to chronological age is strongly predicted by social status in a highly sex specific manner. We don't see that in females, and in fact, we think that's quite informative about the types of experiences these animals can have that are pertinent to the ways in which they age. So with that, I'll just thank my collaborators on this work and on the larger work in the Ambacelli baboons in general, and point out again Rachel Johnston and Jordan Anderson, who were the trainees in my lab who led this work. And I hope there's time for some questions.